Well, hi everyone. My name is David Villar, and it's a pleasure to be sharing this presentation with you. For those that don't know me, I have lived in the country of Colombia for 17 years, till recently. And the reason I became interested on this topic is because the US is probably looked up as holding the highest standards in pesticide regulation. So now that One Health in the world is in everyone's vocabulary, they probably don't realize that delaying taking action on chlorpyrifos in the US also delayed taking similar steps in countries like Colombia. In any case, as you have probably heard, chlorpyrifos has become one of the most controversial pesticides in the US, and there are thousands of papers since it was originally introduced into the marketplace in 1965. So it's not easy to select those that are more relevant for regulatory purposes. And just to put it in perspective, this is a pesticide that has been around before many of us were even born. We're talking of 1965. So as an outline for the talk, we will discuss independent academic reviews of industry-funded studies on chlorpyrifos that has been crucial to grant regulatory approval for decades. We'll go through the epidemiological evidence linking chlorpyrifos exposure to neurodevelopmental deficits in children. We will touch briefly on the socio-economical cost to society and the environment of the exposure to chlorpyrifos and other OPs. We'll go through the timeline of important events related to regulation, and we'll close up with some of the lessons learned from the regulatory history of chlorpyrifos. To begin with, there are at least two independent academic reviews of industry-funded studies that have been used to attain registration approval, so we're going to discuss them. The first one was conducted by a scientist at the Department of Environmental and Occupational Health of the University of Washington in Seattle. It's entitled Flawed Analysis of an Intentional Human Dosing Study and its Impact on Chlorpyrifos Risk Assessment. So this study actually reviews the original one by Colston and colleagues of 1972 that has served as the basis for EPA's risk assessment and market approval since its early days and throughout the 80s and 90s. And EPA and even WHO have quoted this study as a scientific reference, despite it was never actually published. So the original study was meant to find the no observed adverse effect level so that it could be used as a point of departure, if you like, to make recommendations on safe levels of exposure. The design enrolled 16 healthy inmates from a pool of volunteers at a correctional facility in New York, and they were randomly assigned to either a control group or three dose groups exposed to a low, medium, or high dosage of chlorpyrifos. And the endpoints were red blood cell and plasma cholinesterase activities done twice weekly. So here is a schematic diagram of the design, which is not ideal because, as we can see, the treatment dates and the durations of exposure differ among the different treatment groups. And instead of comparing all of them against each other, they only compare each individual group against the control, which is the black line on the top of the diagram. And because they only compare each individual group against the control, they could not identify a graded association between dose and treatment effect, as we will see in a minute. So the way they reported the results, as I wrote here, is as the mean rate of plasma cholinesterase depression for the entire treatment period. And it was not statistically different between the control group and the low and the medium dose groups. And because the middle group was no different from the control, they arbitrarily assigned it as the NOAL. So this flawed analysis was quickly picked up by the scientists at the University of Washington. They stated that the design reduced its power to discern a treatment effect and observe a graded response. They also found that 8 measurements were excluded from the middle group, claiming they were unbalanced data, and that led to the incorrect assignment of the NOAL to the middle group, which they claim is also a form of data falsification. So when they rerun the analysis of the raw data with linear mixed effects regression, they demonstrated a treatment times day effect in all the dosage groups. As we see on these graphs, they plotted the days from the beginning of the treatment on the x-axis against the plasma cholinesterase on the y-axis. And it shows that there is a depression over time in the cholinesterase activity, which is greater for the high dose group, kind of in the middle for the middle group, and not so big, but still statistically different for the low dose group. So really the Colston study could not identify an OL for the plasma cholinesterase inhibition. So what are the implications of not finding an OL? We could somehow summarize them saying that absent an OL, an acceptable exposure level cannot be calculated. Obviously that acceptable exposure level is lower than the one they reported, so we need more studies. And in the meantime, more restrictions should have probably been placed on the pesticide. And if those standards for regulation had been followed, it's likely that a lower incidence of adverse health effects would have happened in children of that era. So really the unanswered question that we should be asking is why a study so critical for risk assessment and protection of public health never received a rigorous review. Now, the second study is another example of why industry-funded studies should at least undergo outside peer review, apart from the internal one that is conducted by EPA. In this case, the name is Safety and Safety Evaluation of Pesticides, Developmental Neurotoxicity of Chlorpyrifos and Chlorpyrifos Methyl. And in this case, it was the European Food Safety Authority that commissioned a detailed evaluation by scientists at a university in Sweden. The good thing about the European Union is that in 2019, a Court of Justice ruled that all pesticide toxicity studies that the European Union authorities rely on should be allowed to be scrutinized by academic scientists. So that's really a good thing. Now, the original study was done in 1998. It was called Developmental Neurotoxicity Study of Chlorpyrifos, administered orally via gavage to presumed pregnant rats. The sponsor was Dow AgroSciences, and the design included 20 liters per dose, and they were given 3 different dose treatments from gestation day 6 until postnatal day 11. And the outcomes in the offspring included general toxicity effects, developmental landmarks, brain morphometrics at day 11 and 65 postnatal, and some neurobehavioral tests to assess for motor activity, auditory startle response, learning and memory functions. So the original study of 1998 was actually published in Toxicological Sciences in the year 2000. As we see, the title is Lack of Selective Developmental Neurotoxicity in Rat Pups from dams treated by gavage with chlorpyrifos. If we look at the abstract, it's clear that the intention is to renew registration approval. At the end of the first sentence, highlighted in yellow, it indicates this is a developmental neurotoxicity study that conformed to US EPA guidelines, 1991 guidelines. And the last sentence of the abstract states that there is no evidence of selective developmental neurotoxicity following the exposure to chlorpyrifos. Now, if you look at this paper, it's extremely long. I would say a little bit tedious to follow. And I would certainly not like to be in the shoes of the EPA people having to go through a dossier of probably thousands of pages. So, as it was expected to happen, there was some level of maternal toxicity on the high dose group with reduction in body weight, feed consumption, some mild clinical signs, but it really didn't affect the pregnancy or the production of the offspring. In the middle and the low dose group, there was no indication of maternal toxicity. Now, if we look at this table, it shows the brain measurements of 9 different brain areas. on those pups at day 11 of age. Highlighted in yellow are the dimensions for the absolute measurements of the pups in the high dose group, and as we can see, they are statistically smaller than the other groups. But because the mothers developed signs of toxicity, these effects were attributed to maternal toxicity. As we see, also there were a few points in blue that were significant at the low and the middle group, but because they performed many statistical tests, they were attributed to simple chance and they were not considered real. Now, the independent reviewers look at this table in a different way, and they found something interesting for the cerebellum in both males and females, which is encircled here on the red squares. And this is actually what they came up with on those pups at 11 days of age. They quote that the proper way to demonstrate the absence of a sensitive target region is to express each brain region relative to the brain weight. So what we see on the right graph is the dose groups on the x-axis and the cerebellum height expressed relative to the brain weight on the y-axis. And we see that in both the low and the middle group, there is a significant reduction of the cerebellum height in both males and females. This reduction also happened in the high dose group, but because it coincided with a similar decrease in brain weight, it was not considered different from the control. So they claim that this U-shaped dose response was consistent with an effect of chlorpyrifos on the cerebellum height at the low and middle dose groups, in which, as I mentioned, there was no evidence of maternal toxicity. To the reviewers, this was evidence that chlorpyrifos is affecting the neurodevelopment of the cerebellum. And in light of these findings, they also argue that they should have looked at the substructures of the cerebellum to see if any in particular was affected. And they also found a couple of issues concerning the design of the experiment. The first one is that positive controls are required to demonstrate proficiency of the lab to perform developmental neurotoxicity studies. And in this case, the test laboratory failed to identify any effects due to lead nitrate that serves as an internal quality control for this type of studies. Now, the other shortcoming had to do with exposure timing. As we can see on the graph, the peak of brain growth in humans is reached at birth, but it takes another 11 days to be attained in rats. So you would really need to find a way to expose those nursing pups to the same concentrations they were being exposed before birth, at least until they reach the day of maximum brain growth, which, as I say, is 11 days. They did actually measure the concentrations of chlorpyrifos in those nursing pups, and as we see on the graph, it was not detectable on days 5 and 11. So what they wrote here at the bottom is that the limited data suggests that the exposure of nursing pups in the developmental neurotoxicity study was far below adequate levels during the most intensive period of brain development. Now, they did not mention any issue concerning cholinesterase activity, so that's something I personally found strange. At the time of this study, EPA was still using cholinesterase as the most sensitive endpoint. And if we look at the table, it shows that the plasma and the red blood cell cholinesterase was inhibited in the DAMs in all the dose groups. If we look at the low dose group, it was even 57% of the control group. I think they probably didn't make any deal out of this because in the fetuses, the cholinesterase was only inhibited in the high dose group. So I wanted to check if this observation was out of line with EPA guidelines that they claimed they were following. And under laboratory animal studies, I highlighted in red here, the low dose is generally considered an OIL for adult and offspring effects. Although if the low dose produces a biologically or a statistically significant increase in response, it should be considered a low IL. So according to these EPA guidelines, not even the low dose group could be considered a no IL. At the most, it could be considered a low IL, as wrote here. And as we just saw in their results, they considered the middle group as the NOAA group. So if we summarize everything they concluded from this review, they say that the NOAA reached by the test laboratory of 0.1 milligram per kilogram per day, which is the middle dose group, was questionable because there were important omissions from the guidelines of good laboratory practices, in particular lack of effects on the normal architecture of the developing brain. There was also a failure of a positive control group to detect any effects, and the design was inappropriate for modeling human exposure during the full period of prenatal brain development. And I would also add to these conclusions that cholinesterase was inhibited in all the dose groups in those dams. So moving on to the next topic, let's now turn the page to those studies that have been a turning point for the fate of chlorpyrifos. If we look at the literature before the turn of the century, there are really no studies that have linked exposure to chlorpyrifos with delayed mental and motor skills in children. If we now look at the literature, there are dozens of papers, and the levels of exposure are well below those that inhibit cholinesterase. So really the biomarker or biological effect that EPA was using all along as the most sensitive endpoint after these studies does no longer hold true anymore. On this slide, we have a few of the most renowned studies that have been conducted by scientists at the Columbia Center for Children's Environmental Health in New York. As I say, they have dozens of publications. Here are just a few of them, and they are all briefly referred to as the Columbia studies. And as we see here, they have been sponsored by NIH, EPA, and some private foundations. So before we look at what they did, we should also know that in 1996, the state of New York passed a pesticide report use for the state. So they were able to generate a baseline description of the type of pesticides and the amounts of pesticides used county by county. And on this map of the state, the red dots represent the thousands of gallons of pesticides used. And we see that the heaviest application was not in the agricultural counties, but in the city of New York itself. And of those pesticides, chlorpyrifos was number one. So they conducted a natural experiment, or that's how they call it, which enrolled pregnant women, 90% of whom were using pesticides in their house. They collected environmental and biological samples during their pregnancy and childhood, and they measured different pesticides and outcomes of neurodevelopmental effects. What we see on this timeline of events is that the experiments began 3 years before EPA finally phased out residential use of chlorpyrifos in 2001. So it was after 2001, it was no longer allowed to be used in spray cans, bombs by pest control operators or any other means of application. About 1/3 of the mothers were enrolled in the study before 2001, and the 2/3 remaining after 2001. So on these graphs, we see that the regulations were quite effective at reducing the exposure to chlorpyrifos. The left one shows the levels of chlorpyrifos measured in personal air samplers that the mothers were wearing during their pregnancy. They basically had a backpack with some type of a filter device that collected air, which was obviously similar to what they were breathing in the house. And on the right graph, it shows the chlorpyrifos levels measured in umbilical cord samples that were collected at the time of delivery. And again, in the mothers that were enrolled in 1999, there were considerably higher levels of chlorpyrifos in umbilical blood. And by the time they got to 2003, 2 years after the regulations came into effect, we see that those levels were non-detectable. Now, if we look at the concentration units in blood, they are expressed in picograms per gram. That's 1,000-fold lower than parts per billion. So just to put it in perspective, we're used to measuring parts per million in acute exposures of animals that are submitted to our diagnostic labs. We may even go down to parts per billion if we want to detect residues or metabolites. But here they are measuring 1,000 times lower than parts per billion, which tells us how sensitive their method must be. And at the same time, for any chemical to be toxic at such low levels, it must be something like an endocrine disruptor, which seems to be the most accepted mechanism for chlorpyrifos nowadays. Now, if we look at the vapor pressure of chlorpyrifos in this example of pesticide vapor pressure by the National Pesticide Information Center, we see that it kind of falls in the medium range. So it is quite likely that when used indoors, enough is going to volatilize to make inhalation as a major route of exposure. Now, with regards to the health effects, the first thing they found is an inverse relationship between umbilical concentrations of chlorpyrifos and birth weight and length. As we see here, for every unit increase in logarithmic concentrations, there was a 67 gram decrease in birth weight and a 0.4 centimeter decrease in length of those babies. In the table, what it shows is that this effect happened in infants that were born prior to 2001, but not for those children that were born after the ban on chlorpyrifos took effect in 2001. Now, the other thing they looked at is the association between chlorpyrifos concentration in umbilical blood with cognitive deficits in both children of 3 years of age and then again at 7 years of age. On these graphs, what they used is a validated intelligence scale for children at 7 years of age. And they report that for each standard deviation increase in umbilical blood concentrations, there was a 2.8 decline in working memory and a 1.4 decline in IQ. So on the x-axis, what we see is the chlorpyrifos levels in picograms per gram, and on the y-axis are those results on performance for working memory on the left, IQ on the middle, and perceptual reasoning on the right graph. So as we see, the slope of those curves tend to go down, meaning that the higher the exposure, the lower performance for each one of those cognitive outcomes. Now, adding to the behavioral data, they provided more support for the association between early life exposure to chlorpyrifos and brain development. In this case, they use MRI, which, as you know, is magnetic resonance imaging that allows you to look inside the brain for any structural changes. And they picked 20 kids between the ages of 6 and 11 who were most highly exposed to chlorpyrifos, at least in the highest quartile of exposure, and they compared them to 20 kids that had no exposure to chlorpyrifos after accounting for sex and age. So what we see here is a sagittal view of the brain. We're looking at the brain from the side, if you like. And what they show is that the prenatal exposure to chlorpyrifos reduced the prefrontal cortex volume. Obviously, those are areas of the brain that are most highly linked to higher cognitive functions, such as working memory, learning, IQ, and so forth. In addition, even though it was a small sample size, there was some suggestion of a dose response. So in this graph, we're just looking at those 20 kids that were most highly exposed to chlorpyrifos. And the higher the exposure, the more different those brains was compared to the control ones. Now, when we talk about the weight of the evidence, there is nothing probably more reassuring than finding other scientists reporting similar findings, or at least pointing in the same direction. So here is a systematic review of 2013 by Chilean authors, and they found adverse effects of OPs on children's neurodevelopment in 26 out of 27 studies that they evaluated. Here is a table of the ranking scheme that they used to assess the strength of each epidemiological study. They basically assign a score to each one of the parameters that we see here on the left, how the exposure was assessed, whether they used validated tests for neurobehavioral studies, whether the epidemiological study was longitudinal, that's when the exposure precedes the outcome. That has a higher weight than, for example, case control studies. Obviously, the number of participants, and finally, how they accounted for confounding variables. So out of the 27 studies, as I highlighted there on the right, the strength of the evidence was high for 11 of them, intermediate for 14, and low for 2 of them. So moving on to the next topic, socioeconomical cost to society and the environment. There are actually several papers that have looked at the socioeconomical cost of shifting those curves on neurological performance. Here they look at the exposure to endocrine disrupting chemicals in the US, and they conduct a population-based disease burden and cost analysis. This paper in particular has different tables for the different endocrine disruptors, and they predict how much is being lost in economic productivity. On this particular table that we see here, they have the OPs, of which chlorpyrifos was number 1. And for each 1.8 million IQ points lost, they estimate losses of $44 billion annually. That's obviously assuming that all birth cohorts are being similarly exposed from one year to the next. So even if for an individual person losing some IQ may not be easily perceived, when we look at a population level, this is obviously a huge unaccounted cost to the economy. Now, the other unaccounted cost of pesticides is the collateral damage on non-target species that we rarely think of when we apply pesticides. I did not prepare anything for chlorpyrifos, but at least wanted to show you this one I made for fipronil, and the list that we see here is probably bigger for chlorpyrifos. So you probably know this already, but just as a reminder, in the middle we see the invertebrates. If we look to the right, we have the largest phylum in the animal kingdom, which are the arthropods. That arthropods comprise millions of species, of which the class insects are probably the largest, obviously. And the references that we have here are different studies reporting toxicity of fipronil at environmentally relevant concentrations for at least some of the species within that order. So the bottom line is that not only may we be killing our target species, but we may also be wiping out all these other guys, and who knows how many indirect effects by messing up a few food webs. With fipronil, just like with chlorpyrifos, probably the most famous non-target species, as you probably know, are the honeybees, which are up here in the order of the Hymenoptera. Now, another unaccounted cost to pesticides is resistance. In the US, they estimate a toll on the agricultural sector of about $10 billion, if not more. What we see on the slide is a healthy specimen of the southern cattle fever tick, which is probably the main obstacle to raising European breeds of cattle in tropical countries like Colombia and Brazil. This tick, depending on its weight, can lay between 3,000 and 5,000 eggs. And as we see on the image, those eggs are hatching and the larvae are beginning to crawl on that petri dish. Now, the funny thing is that I could have just taken that same photo from a tick exposed to chlorpyrifos, any of a number of pyrethroids, amitraz, any ivermectin. This is probably the most multi-drug resistant parasite that we have around the world at the moment, probably next to the barber pole worm in small ruminants. So when it comes to make recommendations to farmers on methods of control, or even teaching students for that matter, we may as well just jump straight into integrated parasite management practices. And as Jon Snow would probably say, there are many different ways to break up the cycle of the tick without relying on pesticides. Unfortunately, most farmers will rather just run out of pesticides before they will listen to you. And that is something I found really frustrating when I worked in Colombia. Now, moving on to the next topic. So where do we stand now? Well, in 2016, EPA revised the Human Health Risk Assessment for registration review. And as we can see on the Federal Register, they propose to revoke all tolerances for residues of chlorpyrifos, which pretty much would imply eliminating all uses of chlorpyrifos in agriculture. They do acknowledge that the Colombia studies are the most relevant evidence for the purpose of chlorpyrifos risk assessment. On this slide, we see that they are referring to those studies with children of 3 years of age. And as we can see, they report odd ratios for mental delay, psychomotor delay, attention disorders, attention deficit hyperactivity disorder. And in that same document, the Scientific Advisory Panel of the EPA concluded that epidemiology and toxicology studies suggest there is evidence for adverse health outcomes associated with chlorpyrifos exposure well below levels that result in 10% red blood cell cholinesterase inhibition. So what really transpired next is quite remarkable. In 2016, the Obama's administration, EPA, did not finalize the ban before Trump came to power. And the first thing that Trump's new administration did was to reverse the decision to ban chlorpyrifos in 2017. So at this point, all the medical scientific community raised their voice of alarm, setting off a new wave of legal challenges. Some states, like California, took matters on their own hand, and they initiated their own ban. And finally, when Biden took office in 2021, EPA finalized the ban 4 years after it should have been implemented. So what we see on this picture is an estimated agricultural use for chlorpyrifos for the year 2019. As we can see, very heavy application in all the Midwest states. This is a figure of the estimated use in million pounds of chlorpyrifos used by year and crop, going from 1991 all the way to 2018. And here is a timeline of events related to registration and use. What is highlighted in yellow are pretty much the dates for the different events that we have discussed throughout the presentation. It starts in 1965 when it was initially introduced in the market. The next one highlighted is the Colston study of 1972 that pretty much served as the flagship study to make risk assessment throughout the 80s and 90s. In the year 2000 is when under the administration of Clinton, it was withdrawn from residential uses. And it was not until 2009 that the company decided to withdraw the Colston study from further consideration to renew its registration. So it's obvious that nobody had actually taken the time to take a closer look at this one study that was flawed and had fooled everybody for so many decades. And in 2016, EPA finally considers to discontinue all the uses of chlorpyrifos, but that is when the new Trump administration stepped in to maintain its registration. It goes on in the table, but basically it was in 2021 that EPA finally revoked all tolerances for use in agricultural crops. In the country of Colombia, that was done 2 years later, following the example of the EPA. And this is the final slide before the conclusions. This is a slide from Earth Justice. Basically what it says is that we got rid of chlorpyrifos, but how about the other OPs? As you can read on the left, EPA already missed its 2022 deadline to finalize evaluation of the remaining OP pesticides. What we see on the table are the different pesticides that are still on the market, and some of the human health effects that have been shown, not only neurological but reproductive, cancer, immunotoxicity, endocrine disruption. As we see on the right of the table, we have the different type of crops in which the other pesticides are also used. And when reading the end of the first sentence, it is awaiting new science data. It seems like science is being used as a weapon to prevent or at least delay taking any action. There is paralysis because there is no analysis. I don't think we can be comfortable with the idea of waiting for decades until all the evidence is overwhelming to take any action. So maybe a precautionary principle should be adopted for some of the other OPs, or maybe the entire group as a whole. I don't really have a clear answer to that question, so I will defer that question to you. Basically, do you think it's past time to take action on the other OPs? And finally, we get to the conclusions. I'm just going to read them quickly, and we'll close the presentation here, and we'll welcome any comment or questions or suggestions that you may have. So the first one is future industry-funded studies should be commissioned via regulatory authorities to avoid any conflict of interest at the test laboratory. The test laboratory should be chosen by authorities based on their demonstrated proficiency in addressing the required endpoints and successfully performing the test required. For chlorpyrifos, we're talking about neurodevelopmental endpoints. Independent scientists with no conflict of interest should be allowed full access to all the documentation from corporate-funded studies. That is, they can no longer be considered confidential business information. Regulatory review boards should include all valid and relevant scientific evidence for policy making. For chlorpyrifos, it was ultimately the epidemiological studies that finally forced the EPA to phase out chlorpyrifos. Therefore, all peer-reviewed and scientific studies with no conflict of interest should be included for regulatory purposes. And finally, political alignment with industry interest must not be allowed by law to override scientific evaluation processes and decisions, as took place when the Trump administration prevented EPA from implementing a ban in 2017. So this is the final slide. I will close up and welcome any questions or comments that you may have. Thank you very much for listening, and goodbye for now.